Okay, so today, this week we're going to be dealing with a technique called Agarose gel electrophoresis. Electrophoresis in general is a technique used to separate biological macromolecules based on their net charge. To do this, usually this means DNA or RNA or proteins. Those are the most common macromolecules that are separated by electrophoresis. Each of these usually carries a net negative charge. Sometimes proteins vary, but DNA and RNA certainly carry a net negative charge, and so they're impelled to move when you apply an electric field to them. The type of electrophoresis that we're dealing with is called agarose gel electrophoresis. Agarose is a carbohydrate that, once it's melted, will form a gel-like matrix when it's cooled, so that you have these long strands of carbohydrate that form this mesh network with pores that run through the matrix. So our slab will be a nice slab of agarose gel that we're going to insert our DNA into. The DNA we're going to use, well, this is all basically to figure out whether or not we got a PCR reaction that worked. So you prepared your PCR reactions last week, and each subgroup used a different primer set. One set of primers was for blue-green algae, one set was for green algae, and one set was for diatomaceous algae. So if we had, say, blue-green algae and only blue-green algae in our samples, those are the only ones that will get a result from this PCR reaction. The people who used the primer set for green algae or diatomaceous algae, those primers wouldn't work with blue-green algae, and so they wouldn't have gotten a positive PCR reaction. So this week's experiment is all about figuring out which kind of broad general algae we have, who got PCR reactions to work. If it was successful, then you should have billions of copies of your gene of interest, and it's that fragment of DNA that we're going to be separating out by electrophoresis on our gel. So as I mentioned, agarose gel is a matrix of carbohydrates with these pores that run through them. We'll insert the DNA into the matrix and then turn on an electric field so that one end of the gel is the negative end and one end is the positive end. DNA carries a net negative charge, so it's going to be pushed towards the positive pole or pulled towards the positive pole, depending on how you want to look at it. And the only way it can make it towards the positive pole is to work its way through this maze-like mesh matrix of the agarose. So when these pieces, first of all, the pieces of DNA, the fragments of DNA that we deal with, are usually measured in length, and that's how we consider their size, is how long are they, and we measure that in the number of base pairs in that fragment. The larger pieces of DNA, the longer they are, the more difficult time they have moving through this mesh matrix of agarose, but the smaller pieces can zip through pretty easily. So in a given period of time, we'll probably let it run for about 45 minutes this week. The short pieces will move the farthest, and the larger pieces will move less distance in that same 45 minutes. So this is kind of a drawing of what your gel or your slab of agarose will look like. Across one end, you'll create these wells. They're like little pockets in which you'll deposit your samples. That hopefully is the DNA from your PCR reactions. We refer to these as wells, and they're part of making the gel. So at one point you will have your agarose melted, you'll pour it into a mold, and you'll place what's referred to as a comb into the apparatus that as the agarose solidifies, creates these open spaces in the gel into which you can deposit your sample. Usually one well is reserved for a DNA standard, and the space below the well here is referred to as a lane. So this would be lane 1, where we have our DNA standard, and then your samples could go in lane 2, lane 3, lane 4, etc. The DNA standard is simply a group of DNA fragments of known length. So we buy them from the manufacturer, and they put all of these fragments of DNA of different lengths into a common solution. And so you just take a sample of that solution and deposit it into this first well for lane 1. When we apply the electrical field, then that DNA is going to be pulled towards the positive end, with the smallest pieces, this is 100 base pairs long, moving the fastest, and the longest pieces, this is 2,000, moving the slowest. It'll barely move at all. Since our fragment is a PCR product, all our fragments should be the same size, so you should only get one band. This is a band. You should only get one band for your sample, and depending on how large it is, it's probably going to be somewhere, I'm just guessing, but it's probably somewhere between 500 and 1,000 base pairs long, so somewhere in here one solid band should appear if your PCR reaction was successful. If your PCR reaction wasn't successful, you won't see any bands in your lane. Oh, I should also mention while I'm here, so we could determine exactly how long a fragment we have. We don't really care for our purposes. This is a yes or no. Did we get a reaction or did we not? Do we have a PCR product or don't we? But we could determine its size based on how far it moves relative to the known fragments. But also we could figure out exactly how much of it we have. So, for example, for these standards that you see on the left here, you load 10 microliters onto the gel. That means you load 10 microliters of this collection of fragments into this well. Then after all is said and done, they know that there's 86 nanograms of the 2,000 base pair fragment. So now you know exactly what the concentration, and you'll see here that the brighter it is, the more of it there is. So based on brightness, you can determine exactly how much DNA you have. Again, we don't particularly care about that, but it is something that can be done with Agarose gel electrophoresis. So we make up Agarose gels as percent solutions. That means X number of grams of Agarose per 100 milliliters. And usually they're fairly low percentages, something like somewhere between 0.5% and 2% gels are common. The higher the percentage of Agarose you use, we refer to that as being a tighter gel. That means the pores through the matrix are smaller. Tight gels are good for separating either very small pieces of DNA or pieces of DNA that don't differ a great deal in length, that are only a few base pairs different in length. They can resolve into separate bands using a tight gel. In addition to the agarose that we add to make the gel itself, we're going to be adding another component called ethidium bromide to the gel. Ethidium bromide is a fairly small molecule that intercalates between base pairs of DNA. So if you look at DNA as a ladder, with the base pairs being the rungs of the ladder, what it means to intercalate is that it slips in between the rungs of the ladder. So right in between the base pairs. Ethidium bromide fluoresces under UV light, so this is how we're going to visualize the DNA. The key to it is that it only fluoresces when it's intercalated between base pairs in double-stranded DNA. So if it's just in the gel itself, you won't see it. But if it's intercalated into DNA, then it will fluoresce and we'll be able to visualize it. So you'll notice here in the standards again, this band we know is DNA because the ethidium bromide is fluorescing brightly, but the ethidium bromide's in the gel right here also, but because it's not intercalated to any DNA here, it's not fluorescing. So the bands will appear to be bright, and the background gel will appear dark. So when you take a sample of your PCR reaction mixture, you're going to mix that with a visible dye, a dye that you can see without the aid of UV light or fluorescence. This dye moves faster than the smallest pieces of DNA, so that you can see it migrate down the gel the same direction that the DNA will migrate, but it's ahead of the smallest pieces of DNA. It moves faster so that you don't run your sample off the end of your gel. You know roughly how far your DNA has moved, because until we put it under UV light, we won't be able to see the DNA. So we usually refer to this dye as a loading dye or a loading buffer. And you'll take a small sample of your PCR reaction mixture and mix it with that loading buffer, and then take the combination and inject it into the wells of your Agarose gel. So it's a pretty simple process today. We're just going to look at whether or not you get a band. If you get a band, then your PCR product, your PCR reaction was successful. That, at the end of the day, will tell us, hopefully, at least what broad category of algae we have. So again, if the blue-green algae primers worked, but the other two different, then we know we have some kind of blue-green algae. We're at least a tiny step closer to figuring out what kind of algae we have. These are unknown samples, so they may be a mixture. So maybe there's some green algae and some diatomaceous algae. Maybe there's all three. We don't know until we run this gel and see what PCR reactions, what primers worked.